Assalamu alaikum. Dear viewers, greetings. In this video, let us discuss about biosynthesis of cell wall and proteins. Interplay between cell wall and proteins. Cell wall are crucial for cell structure and proteins like enzymes and structural proteins and help to maintain and modify them. This interplay ensures the cell wall stays strong and functional. Biosynthesis of cell wall. Bacteria cell wall have a peptidoglycan structure made up of long chain of alternating N-acetylmuramic acid and N-acetylglucosamine sugar. Each NAM has a short pentapeptide chain attached to it. These polysaccharide chains are linked together by these pentapeptides or by interbridge connection. Peptidoglycan synthesis involves multiple steps and occurs both inside and outside the bacterial cell membrane. In Staphylococcus aureus, two carriers participate, uridine diphosphate and bactoprenol. Bactoprenol is a 55-carbon alcohol that attaches to NAM by a pyrophosphate group and moves peptidoglycan components through the hydrophobic membrane. Enzyme in cell wall biosynthesis. Enzymes help to build cell wall components by synthesizing polysaccharide and proteins, which are essential for the cell wall structure and growth. The synthesis of cell wall occur in 8 stages. Uridine diphosphate derivatives of N-acetylmuramic acid and N-acetylglucosamine are synthesized in the cytoplasm. Amino acids are added to the uridine diphosphate NAM to create a pentapeptide chain with the terminal D-alanines added as a dipeptide. ATP is used for peptide bond formation, but tRNA and ribosomes are not involved. The NAM pentapeptide is transferred from uridine diphosphate to bactoprenol phosphate at the membrane. Uridine diphosphate NAC adds NAC to the NAM pentapeptide to form the peptidoglycan repeat unit. If needed, a pentaglycine interbridge is added using glycyl tRNA. The NAM-NAC peptidoglycan repeat unit is transported across the membrane by the bactoprenol pyrophosphate carrier. The repeat unit is attached to the growing peptidoglycan chain to extend it. During the peptidoglycan synthesis, the bactoprenol carrier transport NAM pentapeptide across the membrane. When it returns inside, a phosphate is released, allowing it to pick up another NAM pentapeptide. In E. coli, an interbridge is formed when diaminopimelic acid reacts with the subterminal D-alanine, releasing the terminal D-alanine. ATP is used for this reaction inside the membrane but not outside. Antibiotics like penicillin and bacitracin disrupt this process, weakening the cell wall and causing osmotic. And then transpeptidation. Transpeptidation is a vital process in bacterial cell wall formation where transpeptidase enzyme create crosslinks in the peptidoglycan layer. This occur in 2 steps, forming a covalent intermediate with a donor peptide and then transferring this peptide to an acceptor peptide to form a new crosslink. This process is crucial for cell wall integrity and shape. Transpeptidase, part of the penicillin binding protein family, are targeted by beta-lactam antibiotics to inhibit cell wall synthesis. Biosynthesis of protein. Protein synthesis is the process by which cell produce protein essential for various functions like acting as enzymes, structural components, and hormones. It involves 2 main phases: transcription, where the DNA is copied into messenger RNA, and translation, where the mRNA is used as a template to build proteins at the ribosome. First step is transcription. Initiation. RNA polymerase attached to the gene promoter on the DNA. Elongation. RNA polymerase builds strands of mRNA by adding RNA nucleotides. Termination. The process ends when a termination signal is reached, releasing the mRNA. In eukaryotes, the mRNA undergoes post-transcriptional modifications, capping. A 5' cap is added for stability. Polyoadenylations. A polyo A tail is added to the 3' end. Splicing. Introns are removed and exons are joined to create mature mRNA. Second step is translation. Initiation. Ribosome attached to mRNA and the first tRNA binds to the start codon. Elongation. tRNA brings amino acid to the ribosome. Amino acid link together, forming a polypeptide chain. Termination. Process stop when a stop codon is reached, releasing the completed protein. Components. mRNA delivers a genetic instruction from the DNA. Ribosome, site of protein synthesis. tRNA delivers amino acid to the ribosomes according to mRNA code. Biosynthesis of protein. The double-stranded DNA copies itself by replication. When it copies, the double-stranded DNA converted into single-stranded DNA and produce mRNA by transcription process. The mRNA was processed through ribosome subunit and produced protein by translation process. The produced proteins are literally followed by themselves and carry out to various functions of the cell. Protein folding process. After translation, polypeptides fold into specific 3D structure essential for their function. Primary structure, the linear sequence of amino acid. Secondary structure, local folding into alpha helix and beta sheets stabilized by hydrogen bonds. Tertiary structure, the overall 3D shape formed by the interactions of secondary structure. Quaternary structure, assembly of multiple polypeptide chains into a functional complex. Post-translational modification, proteolysis, cleavage into smaller peptides or amino acids. Phosphorylation, additions of phosphate groups altering protein activity and interactions. Glycosylations, additions of carbohydrate group affecting stability and functions. Significance, these modifications regulates protein activity, localizations and interactions impacting cellular functions. Thank you for watching this video.